I gained a much deeper appreciation for the complexity of life by taking biochemistry. And one of the really early things you learn in biochemistry is the centrality of amino acids. So I'm excited to get into it more here. We're going to talk about the proteinogenic amino acids. What does that mean? These are amino acids that make up proteins in biological organisms. There are 20 of these amino acids that we'll get into in this video. It's helpful to start by talking about the amino acid backbone. This is a general structure of the amino acid backbone, and we have several parts. We have this group over here, which is the amino group. The amino group is why we call these acids amino acids, or at least that's why we have the amino part in the amino acid name. Why do we have the acid part in the name? It's because of this group, which is the carboxylic acid. Together, we have the amino group again and the carboxylic acid group. And that is why together we call these amino acids. We also have our variable R group down here, which we will talk a lot more about as we get into the 20 proteinogenic amino acids. This R group, the fact that it's variable just means that each of the proteinogenic amino acids has a different R group. It isn't an element that's called R or anything. R is just to show that, oh, this actually is different amongst all the different ones that we have. It's a symbolic representation of some general structure that can vary. We also have a hydrogen over here just hanging out. And then we also have our carbon. Let me go ahead and grab another color here real quick. Carbon. This is actually called an alpha carbon, and I can move over here to talk about why we call it an alpha carbon. As we look at the general backbone of a H2N. Okay, as we look at the general backbone structure of an amino acid, we have the different structures like we talked about. And we have the central carbon. Here in the proteinogenic amino acids, it's an alpha carbon. But in other amino acids, it might not necessarily be an alpha carbon. And here's why. Whenever you have the carboxylic acid attached to the carbon in an amino acid, you want to have a good sense of what the different carbons are if it's attached to a chain of carbons. What does that mean? Let's take this as an example. Here we have the carboxylic acid up here connected to a bunch of different carbons down here. And we can situate or notate which kinds of carbons we're looking at by adding a little subscript here. So we have the alpha carbon, the beta carbon, gamma carbon, and it can go all the way down. The amino group can also be attached to different carbons in this chain. So here it's attached to the alpha carbon, but it might actually be attached to, let's say, the beta carbon. So that would be a beta amino acid. But in the proteinogenic amino acids, we always only have that central carbon, and the R group varies, but the carboxylic acid, the amino group, are always attached to just the alpha carbon. So in all 20 proteinogenic amino acids, we're not going to have a structure with a beta carbon or a gamma carbon. It's always just going to be that alpha carbon. But that's why we call it the alpha carbon. Another thing that is really important to point out is that at physiological conditions, the pH of the mostly aqueous environments that our physiological substances are in is 7. So this means that the proteinogenic amino acids are not actually in the neutral form. So if we scroll up, we can see that both the amino group and the carboxylic acid groups are neutral. Neutral, neutral. However, at physiological conditions, they're actually not. So we can redraw the structure here. And if we take a look, we see that there is this hydrogen here, and there's two hydrogens over here in the amino group. But at physiological conditions, it actually is like this. We have the central alpha carbon here. We still have the hydrogen, and we still have the R group, but we actually have 3 hydrogens attached to the nitrogen. So we have 1, 2, 3 hydrogens, and then we have no hydrogens attached to the carboxylic acid group. This is not going to be neutral for us. We're actually going to have a charge on both over here and over here. So we have now a negative charge on the carboxylic acid and a positive charge on the amino group. This will balance out so that the total charge of the proteinogenic amino acid at physiological conditions is going to be neutral overall. And this state is called a Zwitter ion. This is really important to keep in mind. And if we're curious how this actually happens, really it's because the pKa of the different structures here varies. What is pKa? It's a measure of acidity. So the pKa of an amino group is, about 9-ish. It can vary for sure, but for sure it's above 7. And then the pKa, the carbox group, can be around 4, can be lower, can be a little higher. But the key thing is that it's lower than 7. So when we have the pKa value lower than the pH of the surrounding environment, the substance will act like an acid. So in this case, the carboxylic acid will donate the hydrogen that we saw up here, right here, this hydrogen. At physiological conditions, it's going to donate that hydrogen. And we're going to say that the carboxylic acid is deprotonated. However, also at physiological conditions, since the pKa of the amino group is greater than 7, the amino group and the nitrogen is going to be protonated. It's actually going to accept a hydrogen from the surrounding aqueous environment. So here's H2O, which is water. And then it's going to go ahead and accept that. So over here, the hydrogen is going to move over here, and it's going to be protonated. Why is that? Protonated just basically is saying that the amino group is accepting a proton. A hydrogen element or a hydrogen ion, once it loses the electron, is just a proton. That's all that's in the nucleus. So we say that the amino group is protonated. And the inverse happens where once a substance, or really an acid in many cases, loses a hydrogen ion, it's going to be deprotonated. So again, at the neutral physiological conditions, of pH 7, the proteinogenic amino acids are going to be neutral overall, with a positively charged amino group and a negatively charged carboxylic acid group. And this is referred to as the Zwitter ion. As we learn more about amino acids, it's super important to have a good grasp of their stereochemistry. What on earth does that mean? Well, we can actually start by talking about this idea of chirality. I can say that all 20 except for 1 of the proteinogenic amino acids are chiral, and the only one that isn't is glycine. So we can start by going over here and looking at the structure of glycine. Glycine is achiral because it is not chiral. And chirality refers to whether a molecule has something called a superimposable mirror image. What on earth does that mean? If we take a look at the glycine structure, we can see that there are several different structures. We have the alpha carbon right here. We have the amino group right here. We have the carboxylic acid group right here. And then we have two hydrogens. We have hydrogen 1, hydrogen 2. And if we reorient this amino acid in different ways, we can see that if we were to take its mirror image, we would be able to superimpose it on itself. What does that mean? If you take your hands, for example, you have two hands. You have your right hand and your left hand. You can make a mirror image by holding them out to each other. But just because you have the mirror image doesn't mean they're superimposable. Your left and right hand are chiral, because once you have the mirror image, which your hands are mirror images of each other, you can't ever superimpose them on one another. So if you take a look, I'm trying to superimpose them, but here it's on this hand, instead of a thumb, I have the pinky. On this hand, I have that thumb, right? So you can try your best to superimpose them, but that will never happen. In terms of molecules, we can take a look at this illustration to see what that entails for these different kinds of chiral molecules. So I've drawn this picture to show that we have molecule 1 and its mirror image. So here's the mirror. And then we have molecule, let's just call it 1 prime, just to show that this is meant to be the mirror image of molecule 1. And in this image, I want to show that the alpha carbon right here, and then we'll say that this is the R group, the hydrogen, and then we have the carboxylic acid, and then we have the H3N plus. So this is an amino acid general structure for the chiral amino acid. So everything besides glycine. We can see that the molecule 1 prime is not superimposable on molecule 1 because we're looking at it where the carboxylic acid is pointing straight up. The R group in molecule 1 is actually facing towards us. The other structures are facing away from us. And if we want to draw that in a clearer form, we can put dashes and wedges. So when you have a dash, or when you have dashes, it means that the molecule, or the constituent, I guess, is pointing away from you. If you have wedges, it means that the substituent is pointing towards you. So we can clarify that here as well. The wedge for the R group shows that it's pointing towards us. And then the dashes are showing that it's pointing away from us. And then any straight lines mean that that constituent is in the plane of the paper. So just to recap, in molecule 1, we have R group facing towards us, or outside, we can say in front of the plane of the paper, so facing towards us. And then the dashed wedges are behind the plane of the paper, or facing away from us. So with that set up, we can see that even if we take the mirror image, we can't actually superimpose it on molecule 1. And that becomes clearer if, let's say, we rotate molecule 1 this way and we rotate molecule 1 prime this way. That will give us these structures where we have, just to clarify with the dashed wedges, we have the blue, the carboxylic acid, in the plane of the paper. In both cases, we have the R group behind the plane of the paper facing that way. And then we have both of the hydrogens and the amino groups facing towards us outside the plane of the paper. And again, we still have the mirror images. And there's no way you can rotate these so that the green lines up with the green and the red lines up with the red. So if you try to mash these into each other, these are basically just going to not line up. It's not like you're going to have green here, red here. If you were to match them up, you would have red here and green here from the molecule 1 without them ever being green with green and red with red. So that is what we mean by chirality. And it turns out that when you have chiral molecules, you can notate them as being of the L form or the D form. Virtually all of the proteinogenic amino acids are of the L form. What does that mean? That is a sort of arbitrary notation based on the similarity of structures to two molecules, namely glyceraldehyde. So we have L-glyceraldehyde and then D-glyceraldehyde. In L-glyceraldehyde, when we line up the structure in the way right here, we can see that the hydroxyl group is on the left side. And then in D-glyceraldehyde, we can see that the hydroxyl group is on the right side. And for L-glyceraldehyde, we just get that L from the left, and then we just say that instead of being R, it's D-glyceraldehyde when it's on the right side. And when we line up the amino acids to be in line with the arrangement of glyceraldehyde seen here, we have the amino group on the left side here, and then the amino group on the right side here, which we notate as D. And based on this similarity, we just know that the large majority of the proteinogenic amino acids are of the L form. It's important to know that all L amino acids are of an S configuration except for cysteine, which is of an R configuration. So we have the LD configuration, and then we have the SR configuration. What do we mean by SR? We're going to look at cysteine as an example. So we know that cysteine is of the R configuration. How do we know that? Well, when we're determining S and R configuration, we can take a look at the priority of the substituents attached to the alpha carbon. So here we have alpha carbon, as we know, and then we have different priorities of our molecules of our molecule substituents based on the priority of the element. What does that mean? Well, we know that if we look at a periodic table, and I can go ahead and pull one up now. Okay, we have our periodic table now. We can take a look here at the different elements. And when we want to assign priority for S and R, we can look at things like the priority referring to the larger atomic numbers. So in our cysteine molecule, cysteine amino acid, we have different elements. We have carbon, oxygen, nitrogen, hydrogen, and then sulfur. So if we take a look at these, we have different runs. We have sulfur right here, atomic number 16, oxygen, atomic number 8, nitrogen, carbon, and then hydrogen. And if we take a look, excellent. And if we take a look up here at cysteine, we can go ahead and assign these different priorities. So we don't include the alpha carbon in the priorities. We really want to look at the substituents attached to the alpha carbon. So looking at what's immediately connected to the alpha carbon, we have different things like the hydrogen here, we have the carbon here, nitrogen. And if we take a look at the periodic table, the element out of those that has the highest atomic number is nitrogen. So we assign the amino group a priority of 1. And then next we have 3 options. We have the carboxylic acid group, we have the cysteine R group, and then we have the hydrogen. Between carbon and hydrogen on the periodic table, carbon has a higher atomic number, so it has a higher priority. However, we have to determine the tiebreaker between the carboxylic acid substituent and the cysteine R group substituent. So to do that, we look at the elements attached to the carbon. So I can notate that a little bit differently. We have, in this case, 2 oxygens attached to the carbon in the carboxylic acid, and then we have different molecules here that are attached to the carbon. So we have the sulfur, and then we also have hydrogen. So actually, yeah, I should notate it as, yeah, sulfur, hydrogen right here. Cool. So those are the ones that are attached to the carbon. So if we're taking a look at what the priority is between sulfur, hydrogen, and oxygen, we can look again at the periodic table. And the element that has the highest atomic number is sulfur. Between sulfur, oxygen, and hydrogen. So sulfur is going to be the tiebreaker here. And we're going to overall assign the priority of the cysteine R group priority 2. Carbon beats hydrogen. So again, looking at the periodic table, carbon has atomic number 6, hydrogen has atomic number 1. So the carboxylic acid is going to beat hydrogen. So that's going to give us the 4th priority. So to determine R and S configuration, there's really 2 ways. You can either have the lowest priority substituent facing towards you or it facing away from you. The way I remember it best is if you place the lowest atomic number, or rather the lowest priority substituent facing away from you. So that would look like this. We can put up the cysteine alpha carbon here, and then if we want to put the NH3 amino group here, that's a priority number 1 again. And then we can go ahead and put the priority number 2 here, which is going to be the cysteine R group based on the sulfur again. Oh, sorry, that actually looks like a dash was based on the sulfur substituent. Boom. Okay, now we'll go ahead and add the carboxylic acid substituent, which is priority 3. And then last but not least, we can put hydrogen, which is priority 4. So again, this is cysteine with the priorities, and we have it lined up where we're going from priority 1 to priority 2 and then to priority 3, where we have priority 4 facing away from us. When we have it situated like this with the lowest priority substituent facing away from you, basically directly normal to the plane of the paper, not tangential, but normal to the plane of the paper, then we can go ahead and look at how the rest of the priority groups line up. And in this situation, this is going to give us an R configuration, because when we have the lowest priority group facing away from us, if the rest of the priority groups are running in a clockwise direction, then that's going to be R. We could do it the different way, where if the lowest priority group is facing towards us, then depending on the orientation, then we could still determine. So we would know that if hydrogen is facing towards us, the rest would be facing away from us. And that would mean that once we run along the priority groups, it would be counterclockwise, which would also give us R. So it really depends on whether the lowest priority group is facing towards you or away from you. So in this case, cysteine is going to be R. The 20 proteinogenic amino acids are organized here, and they're a bit zoomed out, so they might be hard to see, but I'm going to go ahead and zoom in a second. I do want to start by saying, though, that we have a lot of information here, and truthfully, anyone taking the MCAT needs to know all of this information. You need to know the structures of all 20 proteinogenic amino acids. You want to be able to draw out each individual structure. You also want to know the categories in which the amino acids fall under. And we have 3 broad categories for our amino acid classification, and then some subcategories within those. So we have the nonpolar amino acids, which are boxed in purple here. We have the polar amino acids, which are boxed in blue. And then we have the charged amino acids, which are boxed in pink. Some things that we need to know, of course, for all 20 of these amino acids are things like the structure, the chemical structure, which we talked about. I also want to mention that you do need to know the name. You want to know how to spell the name correctly. And you also want to know the 1-letter abbreviation and the 3-letter abbreviation for each of the amino acids. So we can go ahead and start with the nonpolar ones here, boxed in purple. And within these, we have subcategories based on the different functional groups of the side chains. We have those that have alkyl groups. There's 5 of those. We have one with a thioether functional group, one with a phenyl group, one with an indole group, and then one with a hydride group. For all 20 amino acids, I've drawn a red enclosing shape around the R group. So we can start with proline. Proline has this R group, and it's a pretty unique one among all the 20 amino acids because the R group connects with both the alpha carbon and the amino group. That's unique to proline. And it has the 1-letter abbreviation of P and then the 3-letter abbreviation of PRO. And then you can see all the different abbreviations as I go through them. Here is the structure of alanine, and then we have valine, leucine, isoleucine, which is an isomer of leucine. They have the same chemical formula, but a different structure. And then moving to methionine, which is this structure. It's the only other amino acid with a sulfur in its structure besides cysteine. Then we have phenylalanine, tryptophan, and then glycine, the only achiral amino acid. Those are on our nonpolar ones. And then moving to our polar ones, we have several with alcohol functional groups, one with a thiol functional group, one with a phenol functional group, and then 2 with the amide functional group. So the first one among these polar alcohols is serine, and then we have threonine. We also have cysteine, tyrosine, asparagine, and glutamine. For our final group, we have the charged amino acids. We have negative charged and then positively charged amino acids. The negatively charged ones are boxed in yellow. We have both of these negatively charged amino acids are also carboxylic acids. We have aspartate or aspartic acid, and then we have glutamate or glutamic acid. For our positively charged, we have 3 of those. Each of them has a different functional group. Lysine has an amine functional group. Arginine has a guanidium functional group. And then histidine has an imidazole functional group. One other thing worth noting is that, or there's actually a lot of things worth noting, but just for our purposes, one other thing worth noting is that we have some overlap between different kinds of classifications amongst these groups. What I mean by that is, for example, we have a number, actually I'll just use red. We have a number spread amongst these that are aromatic, meaning they have an aromatic R group, and those include phenylalanine, tryptophan, and tyrosine. we have a ring structure, at least one for all three of these amino acids. And even though they're not all either nonpolar or polar, we have them spread out across those groups. Hope that's helpful.